So, all over here, this small unit we call them as secretory unit of the duct. The secretory unit, that means the functional part. This part is producing the functions. Once this function is there, then we have to throw the production out. Then we have this collecting system, that means it has to be collected here, in between here, temporary storage point, and then it has to be drained through this particular duct chain system. And to transport through this duct chain system, we need some kind of pumping action, because we need to just like throw these things out, kind of pushing things out. Those kind of special cells are there, we call them as myoepithelial cells. That means epithelial cells, they are producing this particular secretions, they are pouring the secretions in an intervening area, hall area, they are collecting it, and then the myoepithelial cells, the muscle cells will, hey, so they are guiding these things to the duct, and duct will be throwing it out. This is the narrow way of, easy way of understanding how this duct is. Now, we will be referring to this particular functional unit, we call this unit as acini or alveoli. So, acini alveoli means if it is coming again and again, this is the functional unit, that means factory, clear. Now, this factory is composed of what? Cells. What kind of cells? The same epithelial cells that we have learned. Now, there are modifications of epithelial cells, which I am talking here as new term as myoepithelial cells, which we did not talk before. So, myoepithelial cell is a modified kind of epithelial cell. They are having some muscles, myofibrils. They are having a capacity to contract, and with the contraction power, myoepithelial cell contract or squeeze this particular epithelium cell, squeeze this particular fluid out, and as well as direct this particular fluid to go into this particular duct channel system and thrown out of the entire system. Clear? So, with this we begin with this particular glandulous things. So, based on the cell, it may be one cell, maybe two cells. We can classify the gland unicellular or multicellular. Likewise, based on this duct, we can classify exocrine, endocrine. Clear? Next, how this particular gland is doing this secretion, we can classify merocrine, holocrine, apocrine. If it is partially throwing this particular thing out, or it is going for through exocytosis, or if this particular entire cell is just like getting washed away after this, we learned this particular thing in 8 plus, 9 plus, 10 plus, blah blah blah, but we are not going to talk about this classification because it makes no sense. Now, this is what we have to know about basis of the glands. Together in summarize like this, number of cells we can classify, presence of the duct and manner of secretion, this is what we know so far. Now, coming to little complex magnification of this classification. The exocrine glands can be called as simple glands and compound gland based on the ducts which they have. If the duct is looking like a single duct, we call them as unbranched duct. If they have more than one branch, we call them as branched duct. So, based on that, we can classify simple or compound. Clear? Now, this is which one is simple? Left one, visibly, right? This one complex, right? So, it is kind of like a better architecture. This is old architecture. This is Windows XP, Windows 10, 12, 14, whatever. Clear? So, this is oldest form. We hardly see these kind of glands. Only in embryonic phase we see these kind of things. Later on, we have to update, get newer versions. So, iPhone keeps on updating and our system do also, right? So, this is how this particular element is classified: simple, compound, branched, unbranched, and how this particular duct look like, we can also classify that: tube like, coiled like, branch tubular, branch acini, etc., etc. So, we can make it complex as many times you want it. So, life is simple or complex based on how we are looking at it, right? So, it is very easy to make our life complex and miserable unless until you make an effort to make it simple. Classification based on secretions. How the secretions are coming, we can classify it. How? If the secretion is sticky or if the secretion is fluid like, right? So, depending on, we can classify it serous secretion, that means it is a watery secretion but high protein. Similarly, there are other kind of secretions are there, we call them mucinous secretion, that means this kind of secretions which is thick, very thick, and it is clear and low in protein. So high in protein, thick, and low in protein, watery, and protein rich kind of things. Now if we see that acini, that means what is acini? Factory, right? Where from the cells are producing the secretions. What kind of cells? These cells? Epithelial cells. Epithelial cells need some kind of modified cells. What are these? Myoepithelial cells. Clear? So this particular acini or factory looks like what? Dark. This factory looks like pale, right? So this kind of acini we call them as differently than that of this. So we call this acini as serous acini. This one is mucinous acini. Because why? This secretions which are present inside is filled of high protein. If you have high protein, they wear very shiny clothes. Alright, all time DJ. And this time bhajan. भगवान के नाम लेते हैं, ऐसे ही रहते हैं. So this is kind of like picking up all the colors. Whatever the dye we are putting it, they are saying alright, we'll wear it, we'll wear it. More fancy clothes, they will take it. And this one is not picking up, नहीं चाहिए. दाल खिचड़ी अच्छा है हमारे लिए. Alright, and this one, more smart guys. Takes both of them. Bhajan भी चाहिए, DJ भी चाहिए. So we call them as Seromucinous, both of them. ठीक है, दिन में भजन, रात में DJ. So this kind of cells, you see that they have a white element here and they have a shiny element over there. See it? So this kind of things we call them as Demilunes. So this is what they are putting a cap or putting a hat. This is a protein secreting cells and they are looking like half of a moon. Alright. So they are called like this. So based on that we can classify it. So this is not what we have to dig in. Whenever you are going to see these slides, we do not have to do these complex things, but you should be knowing what it is. What we will be learning here, what kind of color they are picking. It is DJ or bhajan, that is what we have to pick it up. All the simple things. How do the cell look like, or if it is both of them. So our motto in this class, we are going to pick up the nature of these particular secretions which are present, or what kind of acini we are dealing with, what kind of acini we are seeing it. Mostly what do we see that they are a mix of things. All are smart guys. They do the DJ, they do the bhajan, right? So that's the right thing to do. So the glands which are primarily focused in our class is salivary glands. There are three types of salivary glands. We call them as serous salivary gland, we call them as mucinous salivary gland, and mixed salivary glands. So you understand what do you mean by that? Serous, mucinous. Now explain it to me. What is serous? DJ. Okay. What is mucinous? Bhajan. Okay. And mix. What guy you are? Bhajan guy, DJ guy, or mix guy? Mix guy. Okay, good. Alright, so these are the things which is identifying feature. Now there are, other than this, how these particular factories are fitted with this kind of like sewerage system, like duct. That lumen of the duct, you can look into it. If it is a narrow tunnel or big tunnel. Now if you tell me with your common sense, like if you have a watery fluid and if you have a sticky fluid, so what kind of sewerage you need to drain this fluid out? You have two factories. One factory producing very sticky material, just like chewing gum, and one factory is producing watery milk, right? So you need to create a tunnel to take these things out. So what kind of tunnel you are going to make in? For watery? Narrow. And for the sticky one, you need a wider barrel, right? So that it gets some shape and slowly it will move. Anyway, it is going to be slow and water is going to flow through it, right? So the diameter of lumen is, logically, it is going to be different. In case of this particular like watery one, it will be narrow lumen, and this mucinous, it will be wider one. So if you seeing that particular area of the gland which are having abundance of this particular mucinous, the lumens of that particular like a duct will be more. and if you have this abundance of this particular serous part, that part you will have a thin narrow lumens which is draining these things out. Now, how we are going to see this particular ducts, that is for of concern. So, this is the mixed guy which we are actually, right. So, we have this cap and we have this particular cap which is fitted with the serous one, and rest of the thing is that kind of element. Now, you see that this is how you are going to look into it. So, this is a high magnification, this is not what you are going to see, right, but this is for your understanding. Now, look at this particular stains over here, you try to find out which is brightly stained, which is less brightly stained. You see that not all of them are same, right, they are kind of mixed things are there. So, some of them are deep stained, some of them are pale stained, some of them are having both of them together, right. Now, once you have identified them, then you kind of identify this kind of acini what you are looking at. So, compared to this one is pycnotic, that means all shiny clothes, right. So, this has to be serous or mucinous? Yes. Sure about it? Yes. What kind of guy they are? DJ. Okay. Look at this guy. Paler, right? Bhajan Guy, very sad. So, these are the difference of this particular basic difference, serous and mucinous, right? And this is what we are talking about over here. So, this is pale, and on top of it, its head is there, which is Demilune. That means he is wearing a shiny cap, taking both of them, right? So, they are kind of both of the things to it. So, this particular thing is kind of a mixed kind of element which you are going to see, which normally we are. Sometimes we are sad, sometimes we are happy, sometimes we are elated, sometimes we are super sorry, etc. Sometimes we are lazy. So, this is life, mix of everything. So, this is how we are going to differentiate between serous kind of cells and mucinous kind of cells. So, this is how they are looking like. Why they are serous? Because they have protein rich element which is taking all the colors, and this particular protein rich substance are present into this particular endoplasmic reticulum, Golgi body. They are present plentifully of them, and this particular thing is wrapped into some kind of wrapper, we call them as zymosome granules. The zymosome granules are basically protein packs. Understand packs? Small bundles particles, they are protein particles. These zymosome particles likes color. That color you are throwing to dye it. That is why they are picking it. Whereas this particular mucous cells, they do not have protein substance into it. That is why they are not catching these things up. That is why they are not taking up that much of color. So, this is a basic difference why some is more colorful, some is not. Someone is liking the color, someone nahi chahiye bhai. Understand? Alright. This is about size, acini type, lumen and cells. This is how we are going to talk about and differentiate. So, these basic things we will be looking into it. So, the example of the serous predominant type of gland is parotid gland, whereas mucous predominant gland is sublingual kind of gland. Is predominant, okay, not everything is 100%, it is not like 100%, but predominant, that means you understand 60%, more than 60% we call them at like that. These are other types of cells that you are well familiar with, paracrine and other is known as autocrine cells and signaling pulses. Paracrines means they are producing the substances, but they are working on the neighbor cells. But this particular substance is not liberated into the bloodstream. They are going into the intercellular substance. That means between the cells they are pouring the secretions, and within this particular neighborhood they are being utilized. So, जो खाना बनना है पड़ोसी में ही भाग करके ले जा रहा है. Thing is like that. So, whatever the things are produced, the neighbor cells are taking it up. How they are taking? No blood supply, by diffusions. What do you mean by diffusions? If I light up a scented fragrant kind of things, it smells, the spread of the smell grows on, right. So, this is called a diffusion, slowly going by, right. So, this is slowly encroaching to nearby and it is falling short, not reaching to the far element. Once this particular element is thrown into the blood system, that becomes endocrine. Understand the difference, endocrine and paracrine? Clear. So, neighbor cells are picking up. Autocrine, self-motivating, looking at the mirror, wow, kya dikhne laga hai. So, whatever the secretions are coming up, they are stimulating the cells and doing the actions. So, these are the things which are known as autocrine activations over there, system things are going on. Goblet cells sometimes comes as a short note. You know about goblet cells? 8, 9, 10 is a mucous secreting gland. It is a kind of cell, but it is a gland, because one man army, everything, right? They can do anything and everything over there. They look like this, vast, flat shaped. They have a secretory granules, they have all these things together, and they are capable of doing so many works. And how do they look like? If you see that this particular thing plentifully found in respiratory epithelium and intestinal epithelium. So, when you go to see this particular thing, we will see that they should be looking like this. So, if we are looking at the cells, these are cells. How do the cell look like? Columns, and the columns has hairs in it, right? So, this brush border kind of cells are there. So, brush border epithelium is there, which is tall cells, and the cell nuclei are this one, and resting on this, what do you call them as? Basement membrane or basal lamina, right? Now, they are resting on this, and all of a sudden you see that this is absent. So, this part is this one, what you are looking at, filled up with mucus, and rest of the glands becomes visible. Oh, this is. So, that means this is not the same cell they are, this cell and this cell are different. So, this one which you are seeing here, absent over here, is basically you are looking at this goblet cell. The goblet cell lower half is visible, upper half is not, because they are filled up with mucus, and mucus is not taking up sense, Bhajan Guy. So, they are not going to pick up this. Lower part they are picking up, because this particular endoplasmic reticulum and elements, they are picking up this particular stain. That is why they are seeing the lower part, upper part is invisible, that is why it look like this, right. So, this element is looking like this. Now, this particular thing is present mostly over this particular intestine or respiratory epithelium. They are single cells, but they are called as a like glands, and these are kind of like a misnomer. They are simple columnar kind of cells. They shape like you've seen that, the vase like or cone like structures. They have three parts: basal part, apical part, and mid part, which is cytoplasmic cells. And on HE sense, your apical part is not showing any stains, they are looking like empty. Lower part, the way they are seeing. Why there difference, we understand that. And if you are going to see this entire cell, like we want to see this particular upper part, lower part, you have to add some special stain. Normally, the stain which I am using is known as HE stain, eosine hematoxylin stain. But in case of this particular stain, HE stain is not going to visualize the entire cells. To visualize the entire cell, we need a special cell. We call them as periodic acid-Schiff cells, staining process. In short, we call them as PAS staining. Right? Then you can see the entire cells, right. So, this is how we can classify this particular glands, and these particular things are going on. Functions of the cell, location, as I said, present in small intestine, large intestine, respiratory tract, conjunctiva, and the prime function is produce the secretions and going there. So, they are like this dot 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 like these are the dots which are seeing here is basically upper part of goblet cells. The lower part is present in here. Clear? So, this is about the simple way of identifying it, but what we are going to identify is basically the cells, what kind of cells they are present like this, and what kind of things we are seeing here. We must be able to identify which is serous, which is mucus. That is the job that you have to do. That is this simple thing you have to do, and for this we have to talk so many words. But eventually you have to cut short everything out. Only thing for you is Guy is to identify which is Bhajan Guy, which is DJ Guy, and which is we guys.